Thank you very much for joining us in this first talk from the Innova Webinars 2025. This is an event organized by the members of the InnovaSpec project, a transdisciplinary research project focused on innovations in communication in humans and other animals. This is the first of 4 very interesting talks that will take place over the course of 2 weeks, with speakers from different countries on different themes in communication. Please follow our Instagram profile to get more information so you do not miss any of the talks. They will be amazing. Today, we are very pleased to welcome Dr. Jimena Bernal. She's a full professor of biology at Purdue University, where she's also director of the Research Excellence Access and Learning Scholars Program and has a courtesy appointment at the Department of Entomology. Additionally, she's a research associate at the Smithsonian Tropical Research Institute. Her research focuses on the ecology and evolution of animal communication, particularly in neuron advertisement calls. While neuron advertisement calls are mating signals to attract potential mates, they also draw unintended eavesdroppers, like predators and parasites, a phenomenon whose evolutionary ecology remains poorly understood. Using diverse methods, her lab investigates the behavior and ecology of such interspecific eavesdropping. More recently, she has explored the role of behavior in neuron-invasive success, seeking to understand why some species thrive in new environments despite global amphibian declines. Her key research areas include sensory ecology, species specificity in eavesdropping, frog calls, signal synchronization, anthropogenic effects on communication, and sound traps for mosquito control. Dr. Bernal, thank you very much for your presence here today. After your talk, we will have 15 minutes to take questions from the audience. Everyone can ask questions through the YouTube chat. Questions could be in Portuguese. As perguntas podem ser em português, pessoal. Our team will select through the YouTube chat, and I can translate. With no more details, I will give the word to Dr. Bernal. We'll be here, him and I will be here in the background if you need any assistance or if you have any problems on technical problems.
Erika, muito agradecida pelo convite. Thank you everyone for joining me today. So today I'm going to be talking about how sensory filters and eavesdropping enemies shape novel signal components. And I'll give you a little bit of a flavor of what we do in the lab and how new ornaments evolve and arise. So when we think about animals, it's very obvious to us that oftentimes they have these very elaborate, exaggerated, highly ornamented signals. And across taxonomic groups, males in particular deploy these conspicuous signals during the breeding season. Visual ornaments like the ones on the screen right now are particularly appealing to human observers of these interactions like us. However, ornamented signals are not restricted to the visual realm. Many animals, but most notoriously insects, birds, and neurons, frogs, heavily depend on acoustic ornamented signals. And when we think about how novel traits evolve and appear, we can think about how animals go from producing very simple calls to more complex acoustic signals or more elaborate, brighter visual cues. And these elaborate traits have long fascinated but also puzzled scientists. Darwin, for instance, struggled to understand how ornaments could evolve when they seem to put organisms at increased risk of being attacked by predators. Since Darwin, we have taken large steps towards increasing our understanding of sexual selection. And it is now well recognized that males produce signals that travel through the environment to attract females. And females, in turn, impose selective pressures for more elaborate, conspicuous signals. And they could select for new, novel traits. However, our understanding of these interactions has been mostly focused on these dyadic interactions between a given sender and a single receiver. the target receiver, which are the females. But as Darwin suspected, natural enemies that exploit ornament signals are common. And we call them eavesdroppers, which is when they use information in signals by individuals other than the primary target. And we know that eavesdropping is widespread across taxa and sensory modalities. It's not only it doesn't only occur on acoustic communication; it can occur on any sensory modality. And these eavesdroppers use the signal to detect, locate, and attack the sender, ultimately imposing antagonistic selection on these ornaments. And here we illustrated this communication network approach in this system of frogs. Here's a tumbler of frogs produces a signal. It's intended to females to attract them, but also to competing conspecific males to fight with them. However, there are all these other receivers, non-target receivers, eavesdroppers, that are exploiting the communications system to use that signal to come and eat or bite the frog. And when we think about eavesdroppers, just like their known eavesdropping counterparts, predators, they can be arranged across a continuum from opportunistic to specialized strategies that they use. On one hand, we can have opportunistic eavesdroppers, and they do have very general sensory biologies. They're opportunistic in their behavior, they're really good learners, and they're usually predators or parasites. For them, making a mistake and being attracted to the wrong signal is not very costly. And this is illustrated here by this domestic cat that in the 1960s was the first time that it was experimentally shown that some predators could cue in on the calls of their prey. And on the other extreme, we have the specialized eavesdroppers. And they have sensory adaptations that are finely tuned for this task of exploiting the communication system of their prey. They have stereotype behaviors, they have tight evolutionary relationships with the signalers, and they're usually parasitoids. For them, it's highly costly to make a mistake. And in between, in the middle, we have eavesdropping predators that have a combination of traits. They can have sensory adaptations like a specialist, but they also have the flexibility and the high learning ability of opportunistic eavesdroppers. And they usually respond to a wide variety of host prey cues as a generalist. When we think about eavesdroppers, we can consider the effect that they have on the signals that they exploit. And when we consider current signal features and signaling displays, there's extensive evidence suggesting that intense selective pressures imposed by eavesdroppers had driven the evolution of a broad variety of features and signaling behaviors to conceal the signaler, to escape this exploitation. Some of these strategies can be plastic, as we illustrate here. And in these plastic strategies, it means that when there's perceived predation risk of eavesdropping that is high, the signalers deploy these strategies. And it could include stopping signaling, signal pulsation or cessation, or reducing the signaling rate. So they're like, okay, it's a little risky right now. They signal less. They sometimes reduce the signal intensity. So each signal is produced at a lower intensity. And in some cases, when there is perceived high eavesdropper risk, they switch to a different modality. So like in this case, in the laser wax moths, they stop the production of the acoustic signals, but continue releasing an advertising pheromone when the insectivorous bats are present. In contrast, other strategies are more extreme and include evolutionary responses to eavesdropping. So among those, we illustrate them here in these categories. One option is the fixed signal reduction. So in this case, in populations where the eavesdroppers are present, the signal is absent or reduced. So this is the case of the male Bahamas mosquito fish. And when they are in waters in which there is high abundance of their predators, the big mouth sleepers, they have very subtle or absent ornamentation in the dorsal fin. In contrast, when they live in waters without the predator, they have these elaborate ornamented dorsal fins. There's also the use of private channels, or what we call covert communication. So in these cases, it's using either a range of wavelengths or a range of sounds that are not detected and perceived by the enemy. So this is illustrated here in the northern swordtails. They have ultraviolet ornamentation in their body that is illustrated by these bars. And this ultraviolet signal is not detectable by their main predator, which is the Mexican tetra. Some really neat and interesting cases result in signal loss and re-evolving new signals. And this is the case of the crickets in the Pacific field crickets in Hawaii, in which males have lost the ability to produce the sound in the presence of these Ormia parasitoid flies. And in our population, males have evolved novel sexual acoustic signals. And you will be hearing about this unique and really neat system from Dr. Robin Tingitella, who will be giving one of the talks in this seminar series. And last but not least, in some organisms, there has been a complete switch to signaling in a new sensory modality. So that's the case in these blue-bellied scleropus lizards. They lost their blue bellies, and in some lineages, they have gained an increased intensity of head bowing behavior. So rather than have them blue patch that is highly visible to eavesdroppers, they do signals that include head bowing that are easier to turn on and off depending on predation risk. Okay, so today I want to tell you a little bit about the story of our research program, in which we have looked at how signal exploitation can shape communication systems and modulate the evolution of novel signals and the plasticity. So in our lab, we address this from signal exploitation from many different perspectives: sexual selection, the effect of disease, hearing about diversity. And I'm going to address some of these topics, the ones that are more closely related to novel traits today. So I will not be talking about disease at all. But first, I want to tell you a little bit about the natural history, what the system is, and what do we work on, so you can follow along. So in the natural history, we're going to focus on frog acoustic systems, and in particular, this one on the Tungara frogs that produce the call to attract a mate, and on two eavesdroppers, which are the most common ones in this system: frog-eating bats and frog-biting midges. So let me introduce those players. These are Tungara frogs, Engistomops pustulosus. Many of you in Brazil are probably familiar with these little mud-looking frogs. They're small, browny, but they have beautiful voices. And this is Engistomops pustulosus. They have a very big vocal sac, and this is how their advertisement call sounds like. And here's the first eavesdropper, the frog-biting midges. They are little flies from the family Corytralidae. They're small, about 1 millimeter, and they attack the females, like equivalent to what mosquitoes do to us. Females come and get a blood meal from these flies, from these frogs. So here you can see them coming. They land on the back of these frogs in Tungaras, and they walk all the way to the nostril, and that's where they get a blood meal. And in our first research a long time ago, we actually look at whether they were truly eavesdropping. So to do that, we use sound traps, where we have a speaker and a little fan here to collect any small insects that come attracted by the sound, and they're blow into a collection chamber at the top. So we had one that was playing silence and another one playing Tungara calls. Tung, tung. And this is actually the first trial that we did. I was super excited, but let's look at the data. It looks just the same. So what we actually have found is that the frog calls are not only good for attracting them, but they are required. And that in some areas, like where we work in Panama, these midges are highly abundant, and you can trap about 200 flies with a call, with a speaker playing the calls of a motivated male for about 30 minutes. So they're highly abundant, and not surprisingly, like mosquitoes that bite you and me, they pass on disease. So they pass on a blood parasite, trypanosomes, that we have named in honor of the frogs, Trypanosoma tungarae. But here's the other eavesdropper. So these are frog-eating bats, Trichopsirosis, and this is the work that I have done in collaboration with my academic sister, Rachel Page, who is a permanent staff at the Smithsonian Tropical Research Institute. And just as the flies, they are also eavesdropping on the frogs. So the male frogs are calling to try to attract a female, and the frog-eating bats hear them with those beautiful ears that they have, and they come attracted by the call. And as you can see on the video, they get a yummy meal. But what is really interesting about this system is that these frogs produce different kinds of calls. A male that is calling by himself produces a frequency modulated whine that is illustrated here in panel A and sounds like this. Oops. Oh, hold on a second. But any male can add chocks, the secondary components to a whine. And they can add up to 7 chocks, but that's very rare. They usually add up to 3 chocks. And this is how a whine and 3 chocks sounds like. And that's why they're called tungara, because that's how they sound. So they can dynamically alter call elaboration by plastically changing call rate and call complexity. So these chocks are very different from the structure of the whine. As you can see, they have this stack of harmonics, so they're very complex. And early on, hold on, research performed by Stan Ryan and Mike Ryan look at why are these chocks produced and how can we explain that they don't always do it. And it was surprising because they first tried the female tungara frogs. And when you give a female tungara frog a choice between a whine and chocks, they prefer the complex call, the one with chocks. So if these calls are more attractive, why not to always produce them? Do your best to attract females. Well, what they found is that the frog-eating bats are also preferentially attractive to these complex calls. So when a male is deciding whether to produce a whine, a simple call, or a whine with chocks, they're looking at this trade-off between attracting mates and attracting eavesdropping enemies. And almost 2 decades later, I came into this system and I asked, what about the frog-biting midges? Do they have a preference? And to examine this, we use, once again, those sound traps, and we play simple calls from one trap, complex calls from the other one, with synthetic calls, and we repeated the experiment using natural calls. And what you have here is the number of midges attracted to each one of these traps. And it's very clear that frog-biting midges are preferentially attracted to complex calls. And that was very surprising at this point. The reviewers had a hard time processing this, and they were like, but how? And I was like, how? I don't know how, because I didn't have any idea of how the frog-biting midges were hearing these frog calls. And we're going to come back to that in a little bit. But because of this, we decided to try it in nature. And what we did is with Ikkyo Ihara, who was a postdoc visiting at the Smithsonian, we used these devices that he had created that turn sound into light. So we could place them by a frog that was contained under one of the traps. And with a high-speed video camera, we could record the lighting pattern of the devices and know when a frog was calling, how many chocks they were producing, and so on. And then look at how many midges they were attracting. And what we found is that we suspected that call complexity and call rate, they both increase midge attraction. So now we have a system in which all these different receivers are preferentially attracted to calls with chocks. And this is particularly interesting. If you think about how this trade evolved, but also how the receivers all prefer it, we have these very different receivers, 2 eavesdroppers, the frog-biting midges and the frog-eating bats, and then the target receivers, the females. And they have all converged on their preference for hearing frog calls, tungara frog calls. But not only that, they all prefer complex calls, which is really hard to understand because, as you can imagine, these beasts, they all have very different sensory systems. So we know that tracheups have a very mammalian cochlean-like internal ear, and they're tuning mostly towards high-frequency calls that they use for echolocation and navigating. But they also have a unique trait because they're semi-specialized eavesdropper to hear frog calls. And we think that this matches the chocks. The little frogs, they have these tuning curves that are right into the conspecific call, and they have 2 inner ear organs, the amphibian and basilar papilla, and the basilar papilla happens to be tuned to the chock. But what about the midges? Why would they prefer complex calls, and how we can hear this? So when we ask this question about the chalks, this new component, this complexity, we can ask this question based on a mechanistic answer of how they're hearing, the sensory filters. I'm going to focus on that. But also you could ask it from an ultimate perspective. And at this point, and I will not get into that, but if you have questions, we can talk about it. We think that the chalk has traits that allow bats to maybe um, localize it better. We think that for the females, there are not benefits, that this is due to a sensory exploitation. So the chalk stimulates the BP, where they're more sensitive. And for the flies, we don't know. But overall, we know that chalks are associated with higher density of males. So we have proposed that increased efficiency of attack from the eavesdroppers may have selected for this trait. But going back to how they're hearing, I'm going to tell you the story about how we try to understand. We've been trying to understand how these flies are hearing frog calls. And this is a very interesting question because when you look at insects, ears have evolved almost anywhere, many times independently in their body. So every time in this figure where you see red, that's where ears could be. And many of these are tympanic ears, so they can be anywhere on the body. And when we first started working on these frog-bodied midges, we thought they needed a tympanic ear because they were cueing on these frog calls from very far away. So from what we knew about insect hearing at that point, we thought they needed a tympanic ear like the ones that you and I have. And tympanic ears are relatively easy to evolve in insects because you need a thin membrane that will vibrate with sound, a cavity of air behind that allows it to move, and sensory cells that pass on that information to an integration center. So we first look at those traits with my collaborator, Ron Hoy from Cornell, who wrote the book on insect ears. And this is a frog-bodied midget, if you look at the head. And then if you gently remove it, this is what's behind. And we thought we had those three traits. You can see here a thin membrane. We have a cavity of air behind this prothorax. And we identify chordoneural organs that could transmit that information to the brain. But then we tried to shine a laser to show that this actually was a tympanic ear and it was not behaving as we had expected if it was an ear. So after many years on this search, we decided to go back and look at the antennae because the antennae was well recognized to be good for hearing for mosquitoes and other insects, but only in the near field, very close. And this is what the antennae looks like. This is the Johnston's organ, the second segment at the base, that is a conglomerate of cells that is responding when the flagellum moves, activated by the sound waves. So we're like, okay, well, let's look at the antennae. Maybe there is in the antennae. But to really understand how they were hearing, we needed to understand also how they're using sound. And at that point, recently had been shown that mosquitoes, females and males interact when they're using sound. So we were wondering if the frog-bodied midges use sound for mating too. So this is the work by Priyanka De Silva, former grad student. And what we did is we recorded the wing beats in the near field of males alone, females alone, and then males interacting with females and males interacting with males. And this is what we got. So when we have the male wing beats, they sound like this. This is the female wing beats. So you probably could hear that there are sexual differences in the frequency of the wing beats. The wing beats of the females are lower frequency, which is what we perceive a lower pitch. So they're sexually dimorphic. And what we found when they're interacting together sounds like this. Okay, and now take a minute just to realize how adorable this is. But that was a noisy spectrogram, so I extracted the harmonics. And this is what you see when they're interacting. So the upper harmonics, the third harmonic of the male wing beat and the fourth harmonic of the female wing beats converge. So what we see is that in frog-bodied midges, they use sound in mating. And just as in mosquitoes, which is their group, they have this convergence when females and males are interacting in the swarms. And when males and males are interacting with each other, rather than convergence, we see divergence. And usually the smaller mosquito the smaller male is the one that moves its harmonics away from the other one. So this was really interesting, but what this means is that these frog bite images use the ear for two purposes: for mating, but also for eavesdropping on frogs. And that is a little inconvenient for us. So we look from our system. These are the frog-biting mosquitoes, Uranothenia loyi, and I will argue that these are a much better system for hearing frog calls because we can focus on using frog calls. So first, they were described to bite frog calls, and I suspected by how their antennae looked like that they were not using sound for mating. So what we have done is what we integrate behavior, laser vibrometry, and neurophysiology to understand antennal hearing. So with these frog-biting mosquitoes, we first work, this is the work that was done by Richard Singh, a postdoc in our lab, and we did the behavior work. Basically, we have a speaker on one side playing frog calls, a controlled silent speaker, and we look at what the mosquitoes were doing, and females are more attractive to the area with the frog calls. So we confirmed that in the lab, our colony were eavesdropping on frog calls. And then Bianca Rendon, who at that point was an undergraduate and now is doing her PhD on mosquitoes, look at how they were mating. And what we found is that they were not using sound for mating. The male will patrol, zigzagging around. When he finds a female, he does a little twirl with the leg to engage her, and then they mount and copulate without any use of sound. So this makes this species ideally to really tease apart what you need to have on an ear to eavesdrop on frog calls. And we incorporated, as I mentioned before, laser Doppler vibrometry, and this is work that was led by former postdoc Hoover Pantoja. And we had a mosquito that was immobilized. Here we have the sound that will play and a laser vibrometer that will shine to the tip of the antennae, like we do here. So that was the setup, and when we did that, we can look at the mechanical response. And what we have here is the relative movement of the antennae compared to the air. And basically, these are tuning curves. So this is in red for females and in blue for males. And what we found is that the mechanical response matches the frequencies for the frog call, so they can hear them. But the biomechanical response is the start to hearing. What about the neurophysiological response? Is this passed on to neurons? So to address that, we use extracellular recording. We recorded extracellular compound action potentials at the antennal nerve down here under the JO. And what we found is neural responses that match the frog calls and the pure tones, so we could see what the frequency was. And basically, when we did this, we found that we'll play a barking tree frog, and you can see here a characteristic neural response when we see the spikes matching the barking tree frog call. And the number of spikes increase with the stimulus intensity. So this is proof that they really were using the antennae. So many years later, we realized they are using the antennae. And this is really interesting because we have looked at the variation of the antennae between mosquitoes. So this is work that came out last December, in which we look at the antennae of mosquitoes, Aedes aegypti, that bite us humans, versus the Euloii mosquito that bites frogs. And this is work that we did in collaboration with Hoover, who was my postdoc, and then our collaborators in civil engineering here at Purdue, Adway, Tricanadat, Saket, Tasika, and Pablo Sabateri. And what we did is we used finite element models to describe the antennae. So you can see the male antennae of the Aedes aegypti is like a little Christmas tree. It's super plumose. The females is longer, not as plumose. And here we have the antennae of male and female Euloii that are very similar in structure. And using finite element models, we were able to replicate different versions of these antennae. So what we had is one model in which the antennae was assumed to be a rigid rod, so just a stick. Another one in which we added the segmentation characteristic of that species. And another one in which we incorporated also the little hairs, the sensory cells, the sensory hairs. And then with the models, we could pass frequencies by and look at the tuning and how it was changing between these models. So what you have in blue first is the rigid rod, and you can see all these, how they're matched. So then we have in red the segmented ones. And what you can see is that the peaks are shifting to the actual frequency that is in the dotted line. That is the one at which they're tuned. And then the black one, where we add the hertz, is increased sensitivity. And this is an exception because it's different, but I don't have time to get into that, but we can discuss it later. But basically what we argue here is that the segmentation is in charge of the tuning. And then the hertz increase sensitivity of the ear. And what we find is how sensitive they are to which frequencies depends on what biologically relevant stimuli they have to listen to. So here we have in purple the response of the antennae of Aedes aegypti males, and it matches in this green circle the frequency of the females because they use it for mating. In contrast, in Uloi, the peak is way off. The frequency is 652 here in green, at which their females fly because they don't use it in mating. But when we look at Uloi females, their frequency is matched here in green to the frequency of the frog call, which is way off from where the female of Aedes aegypti has its ear tune because they don't hear frog calls, they bite humans. So this was a really interesting development, and you can read more about it, but we think that this is the beginning of a really cool application where we could use bio-inspired sensors to detect frequencies and look at how noise canceling effects can be used for biodetectors. Okay, but that's the beginning, the flagellum. What happens at the Johnston organ? And what is really neat is that there's tremendous variation in the auditory innervation at the JO. And here we illustrate how the standard representative JO looks like. And this is the one that is in charge of detecting the movement of the flagellum to really hear. So we'll be looking at this efferent auditory innervation across species, and I want to call attention to the basal plate here, that is these efferent neurons in pink, because we were hypothesizing this were going to be important, and they seem to be like important. So we have characterized the JO, the terminal different auditory innervation, these terminals of different species, and the structure of the JO across mosquito species, some that don't bite, don't eavesdrop, and the ones in bold, Culex territans and Uranotenia loia, some that eavesdrop on frog calls. So what we are finding is that there's tremendous variation in JO auditory innervation. And this makes sense because some use it for mating, some use it for foraging in frogs, and we're also looking at some species that don't use their antennae for hearing, nor for mating, nor for foraging. And this is the work that is being developed by my postdocs, non-related Dr. Singhs, Richa and Shilpi Singh. And we're working on this right now, so we are still figuring out what's happening. But in addition to tremendous variation, what we're seeing is that the basal plate innervation, it's found when hearing is important in the species. In males, it is Aegypti that is for mating. In Sabetes, that doesn't use sound. There's no innervation there. And then in the frog biting species, there is an auditory innervation there. We're still collecting data here. So I think this is going to be really interesting. And I want to just give you a teaser for one of the projects that I'm really interested right now, is how these little organisms can hear and localize sound. So you and me, it's easy for us to localize sound. We have big heads, so we can compare the intensity of the sound arriving to our two ears and the time difference between the two ears. So this is what we call ITDs and ILDs. However, if you're little like a mosquito, it's really challenging to localize sound because the two ears are close together, so there's not enough time to use ITDs, and there's no difference in intensity of the two sounds getting to those tiny ears. So it was originally proposed that the JO, each one was inherently directional. And basically because it's moving, and as you're seeing in this video, depending on the movement, it will show directionality. However, Richa has performed these experiments where we remove one antennae and make them localize sound, and also with the two antennae. And what we see is that two antennae are necessary for accurate sound localization. So we think they are using information from the two antennae. And this is work we're doing in collaboration with mechanical engineers Hong Cheng Tao and James Gibbert and neurophysiologist Norman Lee, in which we have been looking at the laser Doppler vibrometry to look at the vibration of the antennae and using neurophysiology to look at how sound is localized. And I think this is going to be really neat, and it hopefully will be coming out in a few months, because I think it's going to change how we think about sound localization. Okay, so with that, last but not least, not least, I want to tell you about signal synchronization, because this is another way in which we think eavesdroppers can modulate signaling strategies. When we communicate, we know that signalers they alternate when they're talking. We do that because it allows the receiver to detect, localize, and discriminate the signals. However, if signalers synchronize their calls, there's a challenge for the receiver. So we have looked at this, looking at why frogs synchronize their calls. There are very few, but at least 20 species that synchronize their calls, and we're looking at the presence effect. And this is an auditory illusion in which if you have two sounds that are produced closely in time, for humans within 50 milliseconds from two different speakers, we perceive them as a single auditory event coming from the location of the first sound, the lead speaker. And this has been described across organisms, in birds, a bunch of insects, and in frogs. So we look at these hook-nosed tree frogs, Meliscicilla, who are call synchronizers. This is a leader and a follower. They usually are produced within 8 milliseconds of each other. And we look at whether they were susceptible to this effect. Because if males are calling right after each other, how are females finding them? So we tested the target receiver, the frog-eating bats, and the frog-biting midges. And what we found is that the eavesdroppers are susceptible to this illusion. So here we see the bats attacking preferentially the leader male, the one that calls first, and the flies, the frog-biting mosquitoes, midges, also attacking the leader more. What about the female frogs? Well, we could not find a preference for the leaders, so we don't have evidence that they are susceptible to this illusion. To look at whether it was due to the setup we were using, we also tried Tungar frog females, which in that species males alternate, and we saw, as it's been found in our frogs, that there is a very striking preference for the leader. So what we propose is that there's been relaxed selection on the receiver, the females, that has avoided this trade-off between the target and non-target receivers and have resulted in what we call a war of attrition. These courses are very, very unusual for frogs. Everyone's silent, and then suddenly one frog calls, and everybody calls, and then they stop. So we think that this has been a way to evade receivers. So a new signaling strategy to avoid receivers. And we have shown this is also true in our systems in which there are called synchronizers, like the Japanese tree frog, in which there are other types of eavesdroppers. So I will argue that by incorporating the eavesdroppers, a previously puzzling phenomenon that was difficult to explain, of why you will evolve a novel signaling strategy, makes a lot more sense. And now for the last part, I want to share with you briefly why we look at biodiversity. And I think I've been focusing on micropredators because that's what we mostly work on. However, if you're interested in how flies and frogs interact, I want to give a shout out to Leonardo de Campos, who has this really neat review in which he looks at the different ways in which flies and frogs interact, and it's just coming out in the annual review of entomology. He has also been using this strategy to broadcast frog calls to increase our understanding of this poorly sampled and unknown taxonomic group. But we've been also interested in looking at how we humans are affecting these fly-frog interactions. So this is work that was first led by Tegan McMahon, and we look in Panama about how do frogs and frog-biting midges respond to urbanization. And what we found is that, not surprisingly, in cities, there's more light, artificial light at night. There's also more anthropogenic noise. But we wonder, what about the frogs? And what we found is that the frogs are everywhere. And actually, they're equally abundant across urban and non-urban areas. However, the frog-biting midges were absent on our transect. And we wonder why. So we experimentally look at this. We did experiments where we had sound traps with artificial light at night, anthropogenic noise, and what we found is the number of midges declines tremendously. Anytime that you add a little bit of light, even zero point 0.1 lux, and as soon as you add noise too. So both sensory pollutants really compromise the ability for us to attract the flies. But how are they affected by these sensory pollutants? Well, to look at that, we put them in an arena where we have males calling and we added the flies and we'll add light or noise. And what we found is that when there's no noise, they do great. These are the number of midges that go to the frog or to the container with the frog, and they could go inside. And as soon as we add noise, even low noise, the number of midges declines tremendously, the ones that can make it to the arena. And as soon as we add light intensity, it declines too. And actually, the only cases of frog body midges getting a blood meal in our experiments happen in this treatment with low light intensity and low noise, and no noise, just background noise. So what we have is that these sensory pollutants really affect the distribution of these species. So to further look at this, Wouter Hafker led this project in which we expanded this across urban and forest sites in the Panama Canal area. And we confirmed that midges, and in this case, we added the whole network, the frog-eating bats, both eavesdroppers are very uncommon or absent in urban areas. So this is interesting, but what we also found is that females tend to be less abundant in urban areas. So the per capita number of females per male, even though the number of males is the same in the curves, is lower in urban areas. So there are less females in the cities. But when we look at higher ornamentation in calls, we see higher ornamentation of calls from urban areas. So they have higher call rate and higher call complexity. They add more chocks. Does it matter? It does to the frogs. Both females from urban environments and rural environments, they both prefer the calls from the males from the city. So we have them preferentially going to the urban calls. Is this a plastic response? Well, we did mesocosmos experiments where we brought frogs from the forest and from the urban areas and have them grow both in the forest or in the urban mesocosmos call there. And we found that they can adjust call rate. But what is really interesting is that when we do look at call complexity, the chocks, in urban environments, the urban males have the ability to increase more chocks, which the rural males, the forest males cannot do. So this suggests that there's been, and thanks to the anthropogenic environment, there's been a shift in this novel component, what is the chock. So what we found is that across this gradient, we have frogs all around, less females per capita in the cities, which had increased the strength of sexual selection, but there's a release from natural selection because the enemies don't happen in cities. So with this, I would like to argue that eavesdroppers are key to understanding the evolution of novel traits and novel strategies, and I think we're going to see more of this happening in the context of how we humans are changing the environment. So this is the whole story for today. I'm very grateful to the great team of people that has worked with me over the years. I have had many amazing collaborators that are field assistants, undergraduate researchers, grad students, postdocs, and our faculty. And I'm grateful for everyone and for the funding sources too that have allowed us to do all this work. And with that, I will be happy to answer any questions.
Hello. Thank you so much, Jimen. Amazing talk. I'll first see if you have more questions here. And if don't, I have a lot of them. So let me see. Okay, we have one. Leonidas asked the question. He said in Portuguese, so I'll translate at the same time. So
Okay.
wait a second. Okay, it's evident that the physical parameters of the sounds are important for the survival and species perpetuation. And in this context, the same parameters are independent of the localization of the frogs, for example, in the land or in the water. Like this calls, these parameters of sounds change according where these frogs are in the environment. If it's a water environment, that the sound propagates differently, or in the land, these things change according to your knowledge.
Okay, so in the frogs that I'm talking about, like these Tunga frogs, they call on water. They actually need water because they have huge vocal sacs, and they call the same word out. And that's mostly true with species of frogs. They have very static calls that they don't vary much where they call. However, what I think this person had in mind is what we call the acoustic adaptation hypothesis. That is the idea that there's been selection for the signals to have structures that are transmitted more efficiently in a given environment. And there's a lot of evidence for this on mostly on birds. That if you look at birds, like the birds that call in forested areas versus open areas, the structure of their signals, the songs varies, and they're very suited for transmission in their own environment. In frogs, however, the evidence is not as strong. And in our study area in Panama, um, a study a while back looked at this and by Nikki Kime and Matt Ryan, and they found no evidence of acoustic adaptation. However, one of my current students, Kathy Gonzalez, has been looking at this again and looking at a broader scale. So she's looking at what about calling in the canopy versus at the ground level.
Mm.
If you're a canopy frog, would your structure be better suited for transmission in the canopy versus at ground level? And what we're finding is that it's complicated. It's not as easy as you would like, and there's not such a perfect match of an adaptation to that specific environment. But we're still looking at that.
Hello. Okay, okay. Thank you, Jimena.
Mhm.
Let me see another. Then the internet froze and I come up. Oh my god. Okay, is everything right now? Okay, next question. Isabella asked, do you think the difference in the ability of producing complex calls in forests and urban male frogs can result in speciation in the future?
That would be so neat. And the answer is we don't know. However, Bouter, who led that, has done an experiment where he brought them um, yeah, he brought them into captivity and he was looking into whether they will mate with each other and looking at the offspring and raising them. And we don't know the answer yet, but it could potentially lead to speciation. The issue right now is that all females prefer complex calls. All females prefer the calls from the city. So we will expect for speciation to really look at local adaptation and that the forest females will prefer the forest calls. But no, the forest females prefer calls from the city. So that will
Mm-hmm.
just mix genetics, but I wouldn't be surprised if something analogous but slightly different could be happening in our systems in which, thanks to anthropogenic change, there's been speciation. And we know very little about that on auditory signals, but on visual signals, we know that pollution in the Tanganyika lakes in Africa has changed the environment and has led to speciation and in some species of fish, in cichlid fish. So it will be really neat if there was something, well, interesting for biologists, maybe not great for the frogs, I don't know. But something like that could be happening.
Exactly. So in this matter, I will ask my own questions, because it's in this comparison between urban and more pristine habitats, because you said that they have few eavesdroppers as flies and predators. So the, there is an effect on fitness of these frogs, for example, number of clutches or longevity, because they are not at the same exposed for the same disease, for example. Do you know if this balance What's happening?
No, that's something that I think will be really neat. Like you will expect that the frogs from the city grow to be older.
Exactly. Yes.
We don't know. We don't know yet. These frogs are very small, but also, as far as we know, they live only for a year. So we expect
Mhm.
survival to be better in the city, but we don't know if it was a longer lived species. We could really fine tune. Like right now, I think it will be really hard to detect differences in longevity, but it will be interesting. We don't know how these frogs are changing. That's something that we're still looking into. Um, Understanding better, and I have an incoming student that I'm very excited about because she wants to go back and look at why are there less females, what is how is this happening, and I even wonder like what are they eating in the city and is
That
this
is
affecting their ability to call, their ability to mate, why are there less females, so we don't know.
spectacular. Okay, we have more, at least three questions. Does its species of frog breeding mages specialize in a specific species of frog, or they are kind of generalists?
So the corytrellids, the frog-bodied midges, seem to be generalist. They do love Tunga frog calls, but honestly, everyone in the forest loves Tunga frog calls. The frog lab calls the Tunga frogs the chicken nuggets of the forest because everybody loves eating them. And when we play Tunga frog calls, we get so many species of frog-bodied midges. And even though they have preferences, they seem to be very generalist. In contrast, the Ranotenia loei, the Ranotenia mosquitoes, they are generalists, but I think in that group we have more specialized mosquitoes. So Priyanka De Silva, who I mentioned, who worked in my lab, she went back to Sri Lanka, where she's from, and there she did this. She found this really neat study, really neat system in which there are three species of Ranotenia, and each one is biting a different species of frog. They're active when that frog is calling in the year, and then during the day, night when they're calling, they're also active at the right height for the frog, and they only go to that frog. So it seems to be a very tight association between those mosquitoes and the frogs there. So it is really interesting, and we're still working on further understanding why in some systems it will be tighter than others.
Nice. Thank you. And during the evolution of these animals, did this sound pattern change in any way, like the frogs and the beaches?
Okay.
Maybe the frogs, I guess. They are talking about eavesdropping, I guess. So this
Can you
frog
can you ask,
sounds.
can you say the question again? I'm sorry.
Did this sound pattern of frogs, I guess, changing in any way along the evolution of these animals? Like,
So do
did
you think pattern refers to synchronization, to the calling pattern?
I guess, or the synchronization or the complexity, maybe.
Okay, well, let me first tell you about synchronization because it is really interesting. So,
Mm-hmm.
people out there, think about how many species of frogs are there. Say, think about a number. Almost everyone gets it wrong. Like people seem to think that there are fewer frogs than there are, but there are about 8,000. It's funny because then you ask people to think about how many species of mosquitoes, and then people always overestimate. But there are many, and out of those 8,000, we know of less than 20 that synchronize, and it has evolved independently many times. And we're looking into why this is the case, but we have sister species in which actually this Smilisca sila is a synchronizer, and it has two closely related species, one that does alternates, which is the standard, and the other one that seems to be in between, sometimes alternating, but the synchronization is not as good. So we know that it has evolved many times, and they evolved from alternating to synchronizing. And what we propose is that it's been done, this change in when you signal the pattern relative to your neighbors is been the result of a high eavesdropping pressure. In terms of the chalks, in that system of angistomobs, other species are other secondary components. Some in Ecuador call them squawks. They sound slightly different. And we know that it's relatively flexible and it has evolved many times. And it's a strategy, a plastic strategy to increase attractiveness to females without always paying the cost.
Okay. And there are two more questions from the audience, both about this thing of urban environment and pristine environment. Do you think the small amount of males in urban environments could affect the genetic variability in that population? Because what do you think?
Yeah, that's a really good point. We know that sexual selection is stronger at the cities, and what that may mean is that mating is biased towards some fewer amount of males. So it will be really interesting to look at the genetic diversity of those populations in the city versus those in the forest. And I don't think that has been examined yet. So that's an excellent point. Yeah. All these good questions.
Nice. Okay, so taking notes also. Diversity. Okay, have you thought about how diseases could affect urban males, seeing as the anthropogenic environment is theoretical more dirty?
Dirty pollutants.
Yeah, exactly.
We have talked about it and I thought about it, but very little. And that's actually the project that my incoming grad student is going to be looking at, where we can detect an effect of pollutants and how the frogs are responding. Every time that there's urbanization and habitat alterations, there are species that are winners, some that can survive.
Mhm.
And in this case, tunga frogs are a survivor. They can make it in the cities. But I wonder at what cost, and whether that is partly what explains why we have fewer females in the city. So I suspect pollutants play a role, but we don't know how or which pollutants at this point. So hopefully in a few
I
years
have a,
we can answer that question.
yeah, I have a tip. I did in my master's. I compare organosomatic index, like relative organ size between an area near a industrial complex in some pristine areas, and
Mhm.
I found that they are not more stressed or don't have problems with body condition, but they presented bigger liver masses and spleen masses and kidney masses. So this trade-off between investment in offspring and investment in organs capable of deal with pollution could be a good answer for these females because females maybe I
That
don't know.
is true.
Just
Yeah, and things can go crazy when we have pollutants. We have looked at cane
exactly.
toads in Florida where they're invasive, and the pollutants from agricultural fields result in intersex males. So we see
Mhm.
males that have ovaries, no way out, but like an eggs. So yeah, unusual stuff happening.
Yeah, I can send you this. This is an
Yeah,
old.
please send it my way. That will be interesting.
Yeah, okay. So I have questions also because I it's really interested your this part of your research on the antennae of mosquitoes because they are dimorphic in some species, and I was wondering about Aedes aegypti because we have a lot of them in Brazil. We have problems every year with it. This year I have dengue. I
Oh,
had dengue, so yeah, we had to deal
I'm
with them.
sorry.
That's okay, but males are more
Plumbus. Hmm.
plumose, so they are more sensitive. And but males do not choose females, right? And males either bite people. Females did. So I didn't
No. So the males don't bite, they live on nectar. They go to the flowers and they live
exactly,
exclusively on nectar.
exactly.
But males, when they form the swarms, males start swarming and they fly in species specific characteristic patterns.
Mm,
Females come to the swarm and males have to be, they're very sensitive because they have to detect the female in the swarm, and then they go court her and then they do the harmonic convergence to the female, and that's when they copulate. So for they have that sensitive antennae because they needed to get to the females in a swarm.
amazing. Because in my logic is thinking about sexual selection, I expected that females are more sensitive to understand and take details on signaling, but it's not the case, right? Because of this perception of females. It's a really nice question, uh, a topic. And the last one, I guess, because the audience, I think they don't have more questions, but I have one more. Sorry. About these mosquitoes. You studied mosquitoes that two species of mosquitoes that bite frogs, but one of them calls like signaling to copulate
Mhm.
males and females, and another one they don't. And there is, in the end, there is more dimorphism, sexual dimorphism in antennae in one species and another. How these things are evolving in these mosquitoes.
We're just starting to learn that. And that's why we're starting to look at a bunch of species across the phylogeny, mosquitoes, and then cortrelates. And we've been mapping the structure of the jail, the structure of the flagellum, and looking at how that can help us understand how they use it. But yeah, in some species, they use it for the two purposes. Some species only for mating, and in other ones only for eavesdropping. In the work that Leonardo has done, Leonardo de Campos, he has shown that eavesdropping on frogs has evolved independently many times, and
Mhm.
I think it's more common that people anticipate in within mosquitoes, within well, actually within flies. So there's a lot to be studied there and to look at map on all this variation in ear structure and what they're doing in real life.
And how these traits are co-opted to both functions, right? To
Exactly.
mating interactions inside the species of mosquitoes and eavesdropping in other species. It's really,
Totally.
really interesting. Okay. Thank you, Jimena. Was an amazing talk. And I guess we finished our questions. And that's
Thank
it.
you so
Thank you a
much.
lot for your presence and your availability.
Muito obrigado.
Muchas gracias para usted. And let me see, boys, can we finish the transmission?